Welcome back, everyone. Chapter 6, we're going to get into talking about the viruses. My personal favorite, I guess, just because that's what I studied in graduate school. So, yeah. So the viruses, so they're not alive, right? Oh yeah. Also, the thing I have to always say, the disclaimer on all videos, which is just that, remember, this isn't everything that can be on the test. This is not everything that can be on. I don't know how many times I can say it, but I feel like people are still going to take the test and be like, this wasn't in the review videos. Yeah, you're right. It wasn't. That's what I will say to you. So anyways, so let's go through the main concepts that were in this chapter. Shall we explain a little bit further on some of these, maybe give a little bit of a refresher for some of you. So chapter 6, these are the viruses. These are not alive. Remember, we consider things to be alive based on cell theory. So they would have to be, made from cells that came from other cells. And these are not made of cells. These, barely have a genome, honestly, if I'm being honest here. So, right, some of them don't even have an envelope. So all viruses are obligate intracellular parasites. Obligate, that term is exactly what it sounds like. Obligated, if you're obligated to do something, you have to do it. These guys are obligated to be intracellular parasites. The rest of this term, intracellular, intra meaning inside, cellular being inside of the cell. Okay, so obligated to be inside of the cell. Parasites are organisms that essentially rely on the host, whatever organism they're living in, for nutrients and cause harm to that host. So we know viruses cause disease in us. That's definitely harmful. These guys, obligate intracellular parasites. All viruses are. So a virion, that's a term for a single virus particle, consists of the genome. So that genome can be single or double-stranded in nature, and they can be RNA or they can be DNA in various orientations. And what I mean by various orientations will make more sense, I promise, after chapter 9. Okay. But basically, positive orientation of the DNA versus negative orientation of the DNA. And that means positive orientation being it codes for the gene directly. You would be able to read the gene from it. Negative being like the anti-copy of that. And you would make another copy in order to make the gene, if that makes sense. So remember, there's two sides to that double helix. One side is the coding strand and the other side isn't. We'll just say that, the template strand, essentially. So one side codes for a thing and the other side will be just the copy of that thing. Remember, A binds with T and C binds with G and all that in DNA. So there's two orientations, 5' to 3' and 3' to 5'. And one of them you read in that direction and the other one is just the copy of it. So the one you read in that direction we call the positive strand. The one that is just the copy is just the negative strand. So they can have negative strand genomes. That means you can't read it directly. You would have to make a copy in order to read it. So that's the thing with a virus. They do these things complicated and have all these different kinds of things. But basically what you need to worry about for this genome, because I know that was a lot all of a sudden, they can have a double-stranded RNA or DNA, or they can have single-stranded RNA or DNA in any orientation. That can be their genome, okay? That's like their chromosome, essentially. They don't have to have DNA. That's what I'm trying to say. Okay, that's their genome. Capsids, they always also have, so they have their genome, and it is usually protected by something called a capsid. A capsid is a protein coating that protects the nucleic acid genome of the the virus. They are made of parts called capsomeres. They're just repeating units, like protein units that come together to make up the whole capsid. That's all it is. So it's just protein pieces. Then we have spikes that are on the outer surface that will help the virus get into the target cells. So all viruses have those three things. The big two being the genome and the capsid, because the spikes can be considered part of the capsid. It can be. Some viruses also have an envelope. That is just a lipid bilayer, a membrane. Where does it get this membrane? It steals it from the host cell. So whenever it is being a stupid little virus cell, and it's got its little particle here, There's that. And then it's trying to get out of the cell. Then next, basically it'll come up to the inside of this and then push through and then pinch off. And then you can imagine that it releases itself and then goes out and can go infect another cell. So that's how budding works. It's like literally when the viruses bud out of the cell, except there will be thousands of them doing it all at once. So they steal the membrane from the host cell. That's their envelope. They can stud the outside of that host cell with their spikes that will help them. These are essentially their keys to get into the next cell. They can put their little spike proteins on the outside of the host cell. And so that whenever they bud out, they are covered in those little spikes that will let them get into the next guy. So those are their spikes. Their spikes are glycoproteins. That's just like a sugar and peptide combination. We also have something called bacteriophages. If you guys haven't seen like the T4 bacteriophages or whatever, they look like alien spaceships. You will have to watch the video if you haven't seen it, but they're nuts. They're pretty nuts, guys. So bacteriophages are viruses that infect bacteria. They essentially inject their genome into the bacteria. They themselves do not go in. They just inject their genome into the bacteria, and the bacteria make more of them, and then they burst out of the bacteria. Always. They always burst out. They don't bud like ours do. So that brings us to the concept of lysogeny and lytic cycles. Basically and we know this as humans, that some viruses can go latent in cells. If you've ever heard of shingles, what happens there is you get infected with the chickenpox, and then later on, whenever you have a less strong immune system, when you get older or you get really stressed out, usually in like past your 50s, it's that same chickenpox virus that you had when you were a kid, the same exact virus, had hidden out into the cells of your nerves and hid out there, and now is breaking free. Essentially, they can hide out in your genome as well. So this is called latency in us, in humans and animals. That's viral latency. We also see that with herpes. So you'll have outbreak and then it'll go away and they'll go hide in your nerves, and then they'll come back along that same nerve that they hid up into and form another outbreak when you start getting stressed or whatever. This is not uncommon in the viral world, right? So that shouldn't be any sort of a shock. So that's called latency. They go latent. In bacterial cells, with bacteriophages, a phage is a virus for bacteria. Basically, like they get infected, the phage injects its genome into the bacteria, and it can incorporate its genome into the bacterial genome. Just stick its little DNA in there. And that is called, a bacteria cell is then lysogenized. And the way that it works is as the bacteria divide and replicate their own genome, they're also replicating the genome of that virus. So all of those new daughter cells that were made from that original infected cell carry that lysogenized bacteriophage DNA in their genome. This can incorporate certain extra genes as well. Sometimes some bacteria can be infected with bacteriophages and get lysogenized this way, have them incorporated into their genome, and it inserts a new gene in there that gives them a toxin or some other thing that helps them survive better. This happens in the case of strep. In Streptococcus pyogenes, which is the causative agent of strep throat, it causes scarlet fever and all this, necrotizing fasciitis. Strep, if it has been lysogenized, can create this toxin called erythrogenic toxin that causes the development of rash, the rash that is associated with scarlet fever. So that's interesting, I feel like, to be aware of. There's actually quite a few cases of things like this going on in bacteria who are pathogenic due to the presence of genes that have been given to them due to being lysogenized by bacteriophages. Pretty interesting. The cooperation going on there. Anyways, so those viruses hide out in the genome. That's called lysogeny. Whenever they are ready to start replicating as viruses again and start, busting out of the bacterial cell and go on to being, viruses, that's the lytic phase, the lytic cycle. Okay, so lytic, normal viral phase, wanting to make new viruses and bust out of the cell. Lysogeny, that's whenever the virus wants to hide out in the cell genome. Okay. What I determined this if you were given the choices between like lytic and lysogeny, I remember it because this has gene in it. I don't know if that helps you at all, but that's how I remember it. Okay. Oh, it even says that about S. pyogenes. I didn't even see it. I guess that tells you how much I've said this stuff over and over again. Okay, oncoviruses. Oncoviruses are viruses that can cause the formation of tumors and cancers. HPV is not the only one that can do that. I know we have mentioned it previously. HPV is human papillomavirus. It is a sexually transmitted infection. It is the most common sexually transmitted infection that there is in the world. Most people will clear it naturally by the time that you have become sexually active. Most people in their lifetime, 90% chance that you've been infected with HPV at some point in your lifetime. 90%, very common. Most of the time you don't know. It can cause genital warts, it can cause cervical cancer, but most of the time people just don't know. This is why the pap smear is so important for women. So you really do need to go and get your pap smears to keep on top of that before it becomes cancerous. Yeah, because why wouldn't you want to get rid of that? So yeah, so those are oncoviruses, the ones that can lead to cancer. They can basically, incorporate themselves into your DNA in a way that messes up your cell cycle and can lead to what we would call immortalization of cells, which means that the cells won't die and they just keep dividing even when they've got all these problems with them. So that's how cancer develops. Overall, viruses are simple. They are non-cellular, right? So they're non-living according to cell theory. They lack organelles. They don't have ribosomes. Sometimes they will steal the host ribosomes and take them with them in the viral particles, but that's uncommon. Another thing that we talked about in this chapter were prions. We talked about a couple of other things too, like viroids and stuff, but prions are important. We will be seeing these again. They are misfolded proteins. We call them infectious proteinaceous particles. They're literally just proteins that aren't folded the way they were supposed to be folded. And because of that, they stick together and form plaques in the brain. And that leads to like the formation of essentially Swiss cheese brain. And we call this a spongy form, like a sponge, encephalopathy. So it is a true encephalitis. It is what we call a subacute encephalitis. Slow forming and takes a long time for symptoms to form, and they're not very striking symptoms. These prion diseases are 100% fatal. They cannot be treated. Treat symptoms, but it's just not very effective even to treat the symptoms. This is what we talk about with mad cow disease. So mad cow disease, Creutzfeldt-Jakob disease is the human version of that. And there's fatal familial insomnia and Kuru and scrapie for sheep and chronic wasting disease in deer. So yeah, that's the prions. So that is it for what we call Unit 1. So Unit 1 is chapters 1 through 6. It's just introducing you guys to the concepts of these microbes. And then Unit 2 is going to be covering basics like genetics, metabolism, and genetic engineering and things like that. So we'll see you guys, I guess, in Unit 2. And remember that for you guys through summer class, that this is going to be Unit 1. There's no test after Unit 1. There will be a test after Unit 2. So Unit 1 and 2 will be the midterm, and it will cover both Unit 1 and 2. Also keep in mind, when we do get to the very, very end of all of this, when we get to the final, and I can't say it enough, is comprehensive. Units 1 through 4. So be surprised when it comes around. So that's that for today. I will catch you guys at, I don't know, I guess chapter 7. I don't even know what, nutrition, I guess. All right.